If you bend a young sapling, it flexes easily in your hands and springs right back. But if you try to bend the trunk of a mature oak tree, you won't move it a single millimeter. A plant's ability to grow rapidly when young, yet withstand hurricane-force winds when mature, comes down to how it organizes its internal architecture. Look at this animation. Notice how identical cells aggregate together to form a single uniform block. In botany, we define a tissue as a group of cells that share a common origin and work together to perform a highly specific function. They lock together like structural bricks to build the plant's organs. To understand the tissues inside a plant, botanists use a single universal rule to classify them: the capacity for cellular division. A cell is either actively dividing or it has stopped. We can map this rule directly onto a master flowchart. All plant tissues split into two definitive branches. First, we have meristematic tissues, which are actively dividing cells. Second, we have permanent tissues, which are non-dividing, mature cells. Pay attention to this split. It is a high-yield concept for your WBCHSE exams. A plant's entire life cycle and structural integrity rely on this exact balance. It needs cells actively dividing to grow and cells permanently locking into specialized roles to survive. Let's compare the anatomy of these two cell types side by side in this T-chart. On the left, the youthful meristematic cell has a very thin cellulose wall, a massive central nucleus to control rapid division, and absolutely no vacuoles. On the right, the adult permanent cell has built a thick cell wall. Its metabolic rate has dropped, and a massive central vacuole has inflated, pushing the cytoplasm to the extreme edges of the cell. Focusing first on meristems, we divide them based on their origin and function. Primary meristems are the original embryonic tissues. They are responsible for a plant's vertical primary growth, meaning they make the plant grow taller and push the roots deeper. This diagram of a shoot apex shows us exactly where these primary meristems live. The apical meristems are located right at the growing tips of the shoots and roots. The intercalary meristems are positioned at the nodes, pushing the stem segments upward. To increase thickness, mature tissues go through a process called dedifferentiation. They revert to a dividing state to become secondary or lateral meristems. As you can see running down the axis of the stem here, these meristems are responsible for the growth of girth. This 3D cross-section of a woody trunk shows lateral meristems in action. The vascular cambium continuously produces new wood on the inside, while the cork cambium generates new bark on the outside. Primary meristems build the plant's initial vertical scaffolding. Secondary meristems arrive later, layering on the immense thickness and strength required to survive for decades. Once cells lose that ability to divide, they enter the permanent tissue category. We break these down into simple tissues, which are composed of only one type of cell, and complex tissues, which use multiple cell types working together, like the xylem and phloem pipelines. Let's do a deep dive into these simple permanent tissues, starting with parenchyma. As this table shows, parenchyma cells are living. They have thin, uniform cellulose walls and feature prominent intercellular spaces between them. This loose packing makes them ideal for storing food and conducting photosynthesis. Next is parenchyma, the tissue of flexible support. These cells are also living, but notice their shape. They pack tightly together with zero intercellular space, and their cell walls are unevenly thickened at the corners by a chemical called pectin. This allows stalks to bend without snapping. Now look at this transverse and longitudinal section of sclerenchyma. These cells stand in strict contrast to the other two. They are completely dead at maturity. They provide rigid mechanical strength because their extremely thick secondary walls are heavily coated in a tough chemical called lignin. Look closely at the longitudinal section on the right. The massive lignin deposition is so thick that it obliterates the internal space of the cell, leaving only this microscopic central cavity called a narrow lumen. Let's test your active recall. Pause the video. Look at these 3 unlabeled cellular cross-sections. Identify parenchyma, collenchyma, and sclerenchyma, and name their defining wall chemical. Ready? A is parenchyma with cellulose, B is collenchyma with pectin, and C is sclerenchyma with lignin. In permanent tissues, structure dictates destiny. The chemical makeup of a cell's wall determines if it serves as flexible support, rigid armor, or soft storage. Now that we've isolated these tissues under a microscope, we have to map them back onto a living organism to see how they collaborate. A plant needs meristems at its tips to grow, parenchyma in its leaves to capture sunlight, collenchyma in its stalks to bend, and complex tissues to carry its water. This diagram of a tomato plant shows how tissue systems are distributed across roots, stems, and leaves. Zooming into the leaf anatomy box, simple parenchymatous mesophyll cells surround the complex xylem and phloem bundles, working together as a whole. Here is your final cheat sheet for the WBCHSSE exam. Take a screenshot now. Remember your direct associations. Pectin links to collenchyma, lignin links to sclerenchyma, and dedifferentiation creates the secondary meristem. Mastering histology means recognizing how these microscopic blueprints dictate whether a plant flexes under pressure or stands its ground. Recognizing these cellular signatures is the key to understanding how every root, stem, and leaf is constructed.